we'll move on to our next speaker, which is Simon Friedman from Northwestern.
Okay. Yeah. Screen sharing okay?
Yeah, all looks good. Take it away.
Great. Thanks. So I'm going to be talking about this central question in developmental biology of how cells change state, make fate decisions, going from pluripotent to some terminal fate. And these state transitions are a lot easier to think about in physics, right? You have some beam with a weight on it, you increase the weight on the beam and the beam buckles, you get some new state. You can write down a differential equation, you can find some critical parameter that is controlling the system. But in biology, we have plenty of state transitions. For example, in development, going from pluripotent, making some cell fate decision, whether to be one type of white blood cell, neutrophil, or another. And maybe there's further state transitions later on that add parts to the neutrophil, build it up. And you might know that there are some important genes, some transcription factors that are sort of telling it where to go. But in reality, when we look at data, we'll find that there's hundreds of genes that are actually changing during one of these transitions and trajectories. And if we start to just write down differential equations for every gene, we'll quickly become overwhelmed. It will be possibly very easy to fit a model, but you'll have no idea whether you're right. So we need some other way of thinking about it. And okay, you'll argue that in the beam, there's also 10 to the 23rd atoms, and we don't care about every individual atom. We care about the collective behavior. We care about the Young's modulus, the coupling between the atoms. And maybe in these gene networks, the gene networks exist, right? There's some coupling between various genes, and genes are acting in concert together. So there's some dimensionality reduction. But how do we get at that and identify critical parameters during a state transition without a generative model? So the framework I'd like to sort of talk about to address that problem is that it's very difficult to get dynamics while you're not changing, but add a bifurcation, add some state change is going to be a lot easier. And so I'll use a one-dimensional model as an example. So just some basic transcritical bifurcation, go from low x to high x as you vary some control parameter a. It's sometimes easier to think about these dynamical systems in terms of like the potential. So if we have some potential that is changing as you go from as you increase A, it switches from going left to right. And if you simulate that with noise, what you'll find is that the variance of data that's exploring such a potential is going to be highest exactly at that bifurcation. And if you look at this movie, it's pretty easy to understand why. Basically, at that bifurcation, that's when this potential is flattest. That's when the curvature is lowest. So this curvature being low is what yields this high variance in the data. And if we only had the data, we could just look at the variance, basically, to find the bifurcation, find the transition. This can be generalized to ND under the framework of continuous time Lyapunov equation. Basically, instead of curvature, we'll talk about the Jacobian. Instead of variance, we'll talk about the covariance matrix. But the statement is very similar to the 1D case. If there's some steady state, if there's some well that we can linearize the dynamics around, that we could say there's a Jacobian guiding, and we're close to that steady state, so the covariance isn't changing very much, then we can write down this equation that relates the Jacobian to the covariance and the diffusion matrix. And generally, this is a very hard equation to solve. We can't necessarily pull the Jacobian out because it's asymmetric, whereas the covariance is symmetric. So it's not going to get us very far in general, except at this bifurcation, because we'll have that flat curvature. Because the smallest eigenvalue of the Jacobian will go to 0, the largest eigenvalue of the covariance will go to infinity. It goes a step further, actually, and you can actually pull out the direction of the bifurcation from the covariance matrix. Those are basically both the modes of the eigenvalue decomposition. So back to biology, right, where we have some big gene expression matrix, and we have a lot of data, but we don't have necessarily any information about the generative dynamics. we're saying that even without a generative model, we can still use the covariance as a function of some control parameter to pinpoint the bifurcation and also determine the key observables, dig into what is driving, what is bifurcating along through this transition. Okay, so that's 1D. Maybe we can do it in higher dimensions in silico. So we drew up this gene regulatory network where gene 1 inhibits gene 2, inhibits gene 1. Write down some equations. This is nice because it exhibits a transcritical bifurcation as a function of this control parameter M1. And we can hook it up to lots of other genes in some noisy system to get a robust sort of covariance matrix that's somewhat realistic of biological systems. And if we measure the covariance eigenvalue as a function of this control parameter, we'll see it very nicely diverges at the same time as the bifurcation. So we have some evidence, and if we're going to compare that, like say, what does it diverge against? Well, we can shuffle the data and recompute this eigenvalue and find that, okay, it's much, much more structure than a statistical null. So from some in silico gene regulatory network, we can easily find the bifurcation. What about real data? So we chose a data set that comprises neutrophil development. This was published last year out of Alona Klein's lab at Harvard. It's a single cell data set with hundreds of thousands of cells and tens of thousands of genes. Each point in this sort of Rorschach is a single cell. It's been reduced to some 2D gene expression space, but in this space, you can cluster the data and label cells as specific types, and you can even connect the dots between various cells and actually get some sort of developmental time trajectory, which they call pseudo-time. So once we have some trajectory like this, we can just compute the covariance as a function of that parameter and do this in bins. And basically, what we found was two things. One was that this covariance increased after about 80 pseudotimes. And two was that we got a nice spike exactly at around 109. So a spike in the data showed seemed to indicate that there was some bifurcative transition over there, and it was well separated from the null. The increase was not well separated from the null. But let's focus on the spike for a second. And what we found was that it actually corresponded very well with this biological transition that was sort of more well characterized, where the neutrophil matures. There are various steps of the neutrophil maturation, but two of them are that it goes from being what's called a promyelocyte to a myelocyte. And when we looked at the genes that characterize these individual states, we found that the promyelocyte genes go down and the myelocyte genes go up exactly at this bifurcation. So that was really encouraging. It seems that this covariance analysis can identify a transcritical bifurcation. I'll talk briefly about the increase. So that seems to line up really well with the peak in the promyelocyte genes. And the reason that's important is because the promyelocyte is the first state after pluripotency in the neutrophil trajectory. So it's the first state after there's a cell fate decision, which might mean that what we have here is actually some sort of phase coexistence. There's maybe multiple states that are sort of swallowed up in our covariance matrix, and what we're seeing is the spread between those various phases. So we can actually go one step further, as I said earlier, not just look at when there's a bifurcation, but sort of how there's a bifurcation. And so in this 1D example, right, the axis of bifurcation, the direction along which things are bifurcating is obviously the only one dimension that you have is x. But in the ND system, it can also be computed as the unstable eigenmode of the Jacobian. And while we generally have no idea what that is, because all we have is basically steady state data, at the bifurcation, we can extract this eigenmode of the Jacobian directly from the covariance matrix. And if you do the math out, what you'll find is that what this means that there are correlations between bifurcating genes at this point of the bifurcation that are going to plus and minus 1. So we can just look at that correlation matrix at the bifurcation, pull out the genes with high correlations, and have a lot more hypotheses for developmental gene regulation. In this case, we found genes that we knew about before, these S100 genes that we knew had to do with neutrophil development, as well as others that sort of fit the criteria. Okay, so just to summarize quickly, there's this question in developmental biology about how cells change fate. There's very nice data that you can get out of these single cell experiments. And I think to connect them, what we found is that there's some physical manifold down there that we can extract using like this continuous time reoptima behavior. We can identify bifurcations, we can find coexistence, we can talk about whether it's easier or hard to reverse the transition to reprogram cells along the Waddington landscape. We can look at the genes that are at the trend that are guiding the bifurcation. I think just more generally, this is motivational, in that we have some evidence for a low-dimensional manifold of development, and that there's physics there, and it would be nice to find out what else is there. So just thank my collaborators and my center, and I'd be happy to take any questions.
Thank you, Simon, for a nice talk. I have a question. So, like related to these covariance, right, that you have on this slide, you showed like related like biological data for these. You had like these blue curves and red curves, right? Can you comment? So, like after you have bifurcations, right, in principle, you can go in two different directions. So there are two possible paths, right? So, is the signal that you measure, is it just going down one of the paths, or is like some mixture of the two paths, right? Because maybe stochasticity can flip you back and forth.
Yeah, great question. So the bifurcation that we're confident about is just a transcritical. So it's going from one state to another. That's okay. The peak. There does also seem to be coexistence. There does also seem to be one of these pitchfork bifurcations, and that's that increase in the eigenvalue. So we actually seem to be seeing both. The coexistence is harder to parse out. It would be clearer if we first like clustered the data. So that's something that we're working on as well.
Right. Okay, so can you then also show the rest of the data on this slide?
Yeah, sure.
Yeah. So here, like you see that first covariance is pretty much 0, and then it starts increasing, and then you have like this spike, right? So at this spike, you said this is where the bifurcation happens. And
that's
like,
where the transcritical, the 1-to-1.
And then what happened earlier when you start getting like the rise of the covariance? Like,
so I think that corresponds to the cell fate decision. Exactly what you were saying, that there's this coexistence between fates. And if you have these coexistence, basically your eigenmode is going to keep spreading out and further and further. And that's what we're seeing in this data. So that seems to be the cell fate decision.
Okay. Thank you. Very nice. So we'll move on to our next speaker, which is Carlos Duque from Max Planck Institute of Molecular Cell Biology and Genetics.